So Jason, what I wanted to ask you about first was, let's just take a time machine back to 2020, 2021. The world was ended, but we were saved by a few vaccines. But around that time, you had, the mRNA vaccine come out. You had a lot of funding for biotech companies. Then you had AlphaFold come out in April, AlphaFold 2 in April 2021, followed by ESM. And we had quite a lot of, hundreds of millions of dollars in funding for a lot of biotechs. And we were like, you know what, we're maybe just 6 months away from ChatGPT for bio. Fast forward 2025, not necessarily the case. We've had some great improvements. We've had a lot of language models come out. But what's happened? The biotech industry is definitely struggling at the moment. So that's what I first wanted to get your thoughts on.
Yeah, love to talk about it. Yeah, I mean, so I think we, so maybe I can give you I'm happy to talk air games sort of on biotech as a whole, but then for this podcast, I think maybe we'll stay focused a lot on AI specifically. Before we jump to AI, I think core challenges with bio have been sort of outside of therapeutics. You haven't had too much repeatable apps that have worked, right? So you had kind of the boom in the 90s of the Monsanto's engineered crops, and that sort of was a pretty solid base of applications since 1982 or whatever, Genentech and insulin. You've had protein therapeutics as a very solid, by far the biggest base of applications, and then at various times you've had attempts at other areas, right? In industrial biotechnology, you had a big boom around biofuels, and you had kind of higher end flavors and fragrances, nutritional stuff, the animal-free meat products. You remember all those different things sort of in sort of synbio space. And if you go back further, you certainly did have industrial enzymes, the Novozymes of the world, and DSMs as a small but solid base of apps. But the newer stuff, I think in 22, you had interest rates go up, and there was a real pullback on venture capital for any of these more out there areas of bio. So that's one of the things that's kind of hurt the community. Certainly those of us that have been pushing new ways of doing biotech, a lot of those new apps kind of fell off. And then in the last year, it's even been tough on the therapeutic side, just because we're now on to 3 or 4 years of tough markets in biotech venture capital for pharmaceuticals. And so that's sort of piling up. And so I think you have that general pressure. Now, how do you fix that? I think you basically, the two levers you have are better tools. And this is what we're going to talk about today, things like AI models and automation and ways that you change the fundamentals, costs and probabilities of success for biotech products. And then the second is regulation, which isn't the topic of this podcast, but I think is actually a big blocker for biotech apps compared to software applications and even things automotive applications and lots of other areas of the market are dramatically less regulated than biotechnology. And some of that makes sense. Therapeutics are designed to perturb human beings. That's a pretty highly regulated space. So we should watch it. But our regulations in agriculture and industrial biotech are pretty nonsensical at this point. So I think there is an opening with new administration, new leadership and regulatory bodies where people are looking and saying, hey, we'd really like to see more biotech products in the United States, and a big blocker is the regulatory. So I'm kind of, somewhat optimistic you could see some changes there outside of therapeutics, especially, but even in therapeutics, as you're seeing with trials on the reg side. But that's maybe as much as we'll talk about all that today. And I think maybe we focus on the tools. Can we see a breakthrough in the tools that develop biotech applications? And then could that really help biotech sort of achieve the latent potential in the space? So does that make sense in terms of the framing?
That makes perfect sense. And we are seeing such an exponential sort of improvement in technology and in tools, especially in sort of with DeepSeek, with OpenAI releasing a new model every day. So there's definitely and then you've got the reasoning models and a lot Anthropic as well. So there's a lot more players in the field, which is great. But yeah, and I think we were somewhat, maybe the biologists weren't expecting that, but the greater community and especially investors, they were sort of expecting a similar trend to be seen with biology. And in some ways, there has been, I've used an ensemble of protein language models, ESM3, Progen 2, OAS, etc. And I've, tried to use this software And I've actually seen results in the lab from sequences that have been generated by some of these language models. So I was like, okay, this is actually, the right mutations are being made and this is actually working. But it's, it's of course not happening, I guess, at the rate that, we expected. Again, personally, biologists didn't expect that, but I'm sure investors and VCs and sort of the greater industry, like I was like, biology is hard. And there is a bit of a rhetoric that, the biology is just like software, just like a big computer. And then we can, throw the English language at it. And, it's not that simple. It's not that straightforward.
Yes. Yeah, I can riff on this a little bit. I mean, so here's like, why do even like the most, like the most aggressive tech people, you still see being pretty enamored by biotech, right? Like, ARC Institute's like funded by the Stripe guys, right? Like, why do they care about biotech, right? Like, pros, what's source? What's that? Yeah.
Project was made by Salesforce. Yeah.
Yeah. And we, ESM came out of Facebook. We, AlphaFold was at Google. We were the first biotech at YC, and that was because Sam Altman took over and was like, we need to be doing biotech at YC. Like, what's up with this, right? And the reason all these people are into it is because, humans are made out of biology, right? So like we're, have our intrinsic kind of interests here. But it's the market for biotech apps is sort of close to infinite. Okay, right? Like it is, like what is the value of adding 2 years of healthy lifespan to people? Right? Like what is the value of, losing 15 pounds for everybody, right? That one we actually know. It turns out it's about $1 trillion market cap between Lilly and Novo. Okay, $1 trillion. One biotech app. Okay, this is not a platform. Okay, this isn't Windows. This isn't AWS. This is one app. Okay, in biotech. All right. So you have this like and that's before you talk about any of the non-human health and wellness stuff, right? Like before you go into all our food is made with biology, right? Like, the environmental impacts, carbon capture, blah, blah, blah. Like go down the list of things, new material, building materials, right? Like physical stuff, right? Like we are made of atoms. We live in a world of atoms, and biotechnology is very much like the programmable atoms-based platform.
Progress your universe.
Yeah, it's the reality. Nothing else is even in the same ballpark, right? Nothing. So that's intuitively true. So it's like, where's all the apps? And like, why aren't they coming out all the time? And it's for the reason you just said, Zishan, which is like, biology's hard. Okay, so all right, so why is biology hard? And the root issue, the number one, very simple reason it is hard for humans is that we did not design it. Okay, so unlike every other engineering discipline, mechanical engineering, I was a chemical engineer, undergrad chemical engineering, computer science, right? Like these are all things designed by humans, built up out of pieces we could understand into incredibly sophisticated objects, an NVIDIA chip, a Boeing airplane, like my God, lots of parts, lots of these, But like you drill into that thing and like down to the lowest level, we understand how that rivet works and how that transistor works. And okay, fine. That's engineering. Okay, you pick up you look at a human cell under the microscope or an E. coli cell, it has like as many parts, like things flying around in there as like a Boeing airplane, but like we didn't design any of it, right? So you're just a manual. 100%. Yeah, you're trying to reverse engineer a object, which frankly is more sophisticated than that chip or that Boeing airplane, right? Just in terms of like the molecular complexity and so on. Like, and that's a tough road to hoe, right? So like that's the problem, right? Like that's why everyone's like, I don't get it. Like, why can't you just like drop some code in? Well, that's the reason, right? Like you can, God bless, it runs on digital code, which is amazing. And since 1982, we could edit that code. And since, the last 10, 15 years, you can, with DNA synthesis, write anything you want and put it in there. And CRISPR, you can put it anywhere you want in any cell type. That's all freaking amazing. Like, but you're running code in a machine that you did not design. And that is a hard problem. Okay. And so that's our root problem. Right. And so then the question is, how do you solve that problem? All right. How do you solve that problem? Because again, the latent potential of programming cells is infinite market cap, infinite human potential, like all these, like, it's crazy, right? Look out the window at what biology does in the natural environment. It's bananas, right? That's just the stuff that was just, produced by random chance and evolution over the last 4 billion years. And it's just incredible complexity. So like, how do we unlock it? Right. And, and so I can talk about that, but does that make sense? Like that's like if you really want to know what's wrong, but like, that's the hard problem. Right. And so if you want to see, you're a young person, people getting in this field today, like that's what's cool to figure out. Right. Like if you can solve the problem, right, of like, how do we design biology predictably, you win all the prizes, right? Like, like there's just no, it makes, the tech industry basically runs on ads and information transfer. It's a small industry in comparison to what biotech could do.
You'll be placed number one in the biotech.
There's no comparisons, right? Like, so we have this obvious thing that if you could just solve the engineering problem, like the potential is infinite. And so, all right, so what should people work on if they care about that engineering problem, right? And I think there's like two halves. You can either work on basically letting you try more designs and generate more data about those DNA designs per dollar. So like you're basically better and better at the the cost for you to try things is lower. Okay. Which lets you try more things per research dollar, which is where you like
was the highest cost in biotech, right? Any single assay or machines, 5 assays can take 10 machines, which can amount to like a couple $1 million here and there. So that trial and error phase in itself, insanely capital intensive.
Yeah. And by the way, other disciplines are like this, like semiconductors is not cheap to try a semiconductor design. So how do they get around it? Well, they simulate it. Okay, so this brings me to the other side of our equation. The other way you can attack this problem, other than like lowering the cost to effectively prototype and test a DNA design, is to be better at simulating the performance of said DNA design, which is a long way of saying understanding what the hell is going on inside a cell. Okay, right? Like biologists, right? Like the work of understanding biology is sort of on its way. If I wear my genetic engineer, DNA designer hat, that's the work of marching us towards simulation. Okay, right? Like, and by the way, that march, I just want to be clear, is much harder than it was for Cadence or, any of these like semiconductor lab people, because we didn't design the pieces, right? So we're sort of like trying to generate the data sets of a system we didn't understand to ultimately be able to simulate the performance of our designs. Those are the two routes of attack. So if you want to make biology designable, either improve our simulators or reduce the cost of prototyping and assaying the performance of prototypes, those are your two roads.
You have to boil it down to two. Okay.
Yeah, and by the way, they can connect. Okay, right? Like cheaper designs, generate more data for the simulator, blah blah blah. But they are kind of like two roads. And I think both of them are fruitful, and both of them need fucking brilliant people working on them, and it is a good use of people's life energy to work on that. Both those problems. And this is all, I've not made one mention about an app here, Zishan, right? So I'm not, I also think like the whole reason to build these tools is that, then someday someone will be able to invent that treatment for the disease that currently doesn't have one, or that thing that makes trees grow faster to take more carbon out of the air, or that thing that develops a new material for Department of Defense, like whatever, like all those apps are downstream of solving the tools problem. And the tools problem has these two buckets: prototype and testing cheaper, or simulators better.
Yeah, the apps come in the second or third.
AI's got to roll in both of those, by the way. But that's how I see the world.
So why can't we? I remember this was probably a Reddit forum. But why can't we just train like as you said earlier, 1 million PhDs that are, once we get AGI or like towards our goal for AGI, ASI, etc., be like, hey, this is all of literature. This AI acts as an expert PhD student, equivalent to someone who did their PhD at your Oxford or MIT, has been working in this for 30 years. Why not just simple, right?
Yeah, sure. So let's like nerd out on like AI for a minute, on like where it could play in this world of like improving the tooling to develop the apps, which I think is the big unlock for biotech, period. So there's two big buckets for AI. One is the one that kind of you were mentioning earlier, that got started in whatever 21, like that where there's this sort of boom around like things like protein models, like AlphaFold, right? And the idea here was basically like there's large neural nets have proven to do amazing things when given a combination of a lot of data to work with and a lot of compute to train on that data, especially with like unsupervised learning. And so, can we repeat that with large biological data sets? And I think ESM is a great example where it's like, almost like for like with kind of like a human language based model, right? It's just training on the sequences. And so like I think that like it turns out, yes, like kind of amazing, right? Like you could really do some amazing stuff in proteins with that. And so that sort of served as like a flag out there that said, if you collect enough data about a certain like category of biology, maybe you could have essentially a simulator, a CAD tool, a design tool, which is really like a little bit what AlphaFold is, right? Yes, it's not doing it's not a physics simulator, okay, right? It's not like old school chemistry, like a Schrodinger kind of like chemistry, it's not, you're not trying to be physics sim, but like you give it a sequence and it predicts, it essentially simulates like what the structure of that is, and there you go. Okay, fine. Okay, so and you're all in silico. So could we do that for other things? That's the question. And then how much better could you make those protein ones since they were already like proving to work? And so you have like a bunch of energy there, ESM, blah, blah, blah. And like, I think like, it's sort of like, oh, is there going to be like a ChatGPT breakthrough? And to some degree, like AlphaFold was that, frankly, like, like getting a Nobel Prize, like, right. Like, so that does show that it works. The issue but what people are really saying when they want the ChatGPT breakthrough is like, show me the money, right? Like I would also like to know that this breakthrough translates into like some big value gain.
We knew that Ozempic too.
Well, yeah, and like this is where like we got a little bit beat up because like the biotech industry does not really have a mature like platform tools ecosystem where like you can come up with some great new tool and drop into it. Like you see, like I want to make the cursor of whatever, right? Like good luck, right? Like that stuff in bio is like brutal, right? We just don't have that. Like this is more like if you're an entrepreneur on the podcast, but like, breaking in there is just harder in biotech than it is in tech for a whole variety of reasons. And so, but like that, so we don't, you don't see as much of like, you haven't seen those models like turn into a fountain of money quickly, for all the reasons. And so that's what people say when they're like, well, we haven't seen that ChatGPT. That's true. And that's why you're not seeing like, some explosion of capital like you see in the human language model space. Okay. Right. But hey, it worked. Like AlphaFold, like that worked. My God. Right. So
People understand how like insane that is, like science-wise.
right. Like bring it back to the tool side. Hell yeah, that is a great line to march down. Like we should be go like for example, we put out a like an mRNA model, right? Like that was trained on a bunch of mRNA data we generated with we have a lot of automated infrastructure, right? So we generate a bunch of data on this. We showed that you could train a model and the model could then predict mRNA stability. Awesome, right? Like let's like keep going. Like whatever aspects people wanted to go after, I think you can kind of take the approach of like throw lots and lots of data at it and neural nets, and you could get there at some level. Okay, right? And the quality is going to depend, and how there's caveats, but like that's not a bad way. It's not a bad line of attack for like simulators, basically. Does that make sense?
Yeah, that makes sense. And
But when it'll convert into value is a little bit about like when people show they're making valuable apps coming out of these things, and that that just takes some time, right? So I do think it's a great area for people to get involved in. And Ginkgo's launched a thing we call Datapoints, which is basically a service to generate those kind of data sets for the people building those models. So if there's some like model area you want to give your startup, and you're like, yeah, we're gonna like nail this, right? Like we're going to nail promoter design for expression in different cell types or whatever, then like we could generate a bunch of data in that direction, and you don't have to build the lab, you don't have to have all the robots, and then you own all that data and you can build your model like that. We're sort of trying to support that community with like lab infrastructure for anyone that wants to go down that road. And we have a couple services that are like more standard because the pharma companies keep asking us for them. But if you were like a startup that wanted to try it in a new area, we're also happy to like just take requests about like where people in the industry want to see more data. And then we just post, we like when we do it in an area, we'll post like a free data set to download. So we've got a few of those up online.
Yeah, I've seen a few of those, and I link those to the show notes that I put up later as well.
Yeah, the one we had, we do like a service with functional genomics, where basically like we will perturb cells either with like a small molecule compound library or with CRISPR edits. So you're like, okay, I want to make these 1,000 CRISPR edits in this, you know, iPSC derived neural cell line or something, right? Like, and we're like, okay, we're going to make all those edits, and then we're going to do a drug seek. This was like a technique that was like pioneered at Novartis, and like basically give you back a big transcriptomic readout on that edit, and then the next edit, and then the next edit, and then you're going to get these big transcriptomic sets, and you can use that for making models for like target discovery, for example. But we have like a cool data set, like 4 cell lines, 15. You can also 15 compounds in this case, and then different concentrations of those drugs, and then a drug seek readout. It's like a terabyte of data that you can just download.
Oh, that's great. It's just like a sort of a digital blueprint that can be ingested by a machine learning model and fine-tuned, etc.
Yeah, if you're like an AI ML person, you want to start like getting your arms around some like actual real bio data with like drug compounds and concentrations in a bunch of cell types and see what you could figure out. Like, it's just there. Yeah.
And another thing, actually, I do want to go a bit deeper in this, but before I get to that, we talked about, eliminating data bottlenecks and whatnot. But where do you see some of like the biggest gaps right now? You mentioned functional genomics, protein design, antibody developability. Like, where do you see sort of the greatest opportunity and unmet need in data development, I guess? And also experimentally verified data. Yeah.
Yeah. So like what we hear from people, like, customers and stuff. There's a bunch of public data on like cancer cell lines. There's like there's a CCI thing, Broad had the depth map stuff. There's a lot of like cancer cell lines. Once you move into other cell types relevant for other diseases, like say that neural cell line I was mentioning, like they're just not the equivalent data sets. So that's one gap. Like, it would be making more of these large, I think, some of the easier stuff to scale is the transcriptomic stuff, right? Like data sets, particularly perturbed data sets, either genetically or with compounds, and then just having those available. Like I think all the disease foundations, for example, should basically be generating big, pay a few $1,000,000, make a giant data set and put it up online and let a bunch of ML and AI people like hunt for new targets for your disease. Like that's like a no-brainer, right? Like everybody should be doing that now because the modeling community has so many tools to work with that they just had the data to play with. And biology is pretty different, right? So you kind of want, it's not enough just to have like a data set on a human cell line and then you're done. It's like, no, I need it on like this I need it on, whatever, like a skin. Cell lines act differently. So you do want to kind of create these data sets for the one that's relevant in your disease area as one example, right? Like, so I think that gap of is a big data set available, especially publicly available in an area that's important to my end application. If I'm, like whatever, like I think you would love to check that box for more and more areas of biology. Like I think that would really help on like the modeling and simulation side of the house with the AI model. So that's the gap I see on the drug development side. So like take antibodies. So the other service we offer, like off the rack, in Ginkgo Datapoints, is antibody developability. So you've got so a lot of people have worked on like binding, for example. Like there's a lot of people working on like binding assays not binding assays, but binding models. All right.
Yeah.
What's that?
Binding affinity, I guess, is sort of the main, I guess.
Exactly. Yes. Can I predict it's more or less like if you're not a drug, discovery person, it's like here's a disease. I have a particular target, like a protein involved in that disease that I want to hit with something, and so I'm going to try to design an antibody that'll stick to it. And maybe the antibody itself is the drug, or maybe the antibody is conjugated to a small molecule, and the combination of the two is the drug. But either way, I need it to like hit that target in the disease cell. Yeah, stick to it. I need it to bind. Okay, right. And so that problem, there's actually a lot of like, there's a ton of lab techniques to do that, ranging from just like hitting an animal model with the target and having its own immune system generate a bunch of antibodies and then taking the antibodies out and sequencing them, right? To phage display, to whatever. Companies like Animab are just built on doing this as a service. So there's sort of a lot of that already out there. And so people have worked on AI models with a lot of data, but it's less of like a gap in the market, right? Like, cause you can already get it. Like if you need a binder, you can get a binder, right? Like, so where there's a gap is like, okay, now I want to, I these 50 stuck to my target. All right. And I want to see like, are any of them going to make for a good drug? Okay. And being a drug has more features to it than just sticking to the target. Like it also needs to be soluble so that like when you inject it in the patient, it doesn't just clump up and go nowhere, right? Like there's just like other features.
Yeah, thermal stability, aggregation propensity, all of that.
Bingo, all the things. Okay. And so there's like about like in the industry, maybe like 10 kind of developability characteristics assays that are like commonly used for these sort of like biological measurements about your drug to see if it's good, right? Before you make the big leap to going ahead and putting it into an animal trial and then into a human, right? So okay, so that part right now, like you could not do easily at high throughput, like, or you could not previously. We have built out a bunch of infrastructure at Ginkgo to run all that on robotics. And so now you could send us, you could say, hey, Jason, here's 1,000 or here's 10,000 antibody sequences. We'll get those, we'll either make them ourselves or get those made, make you the antibodies. And then we're going to put them in and run like 10 developability assays on them. Again, it's like fragmentation, aggregation, heparin bind, poly, thermostability, self-interaction, hydrophobicity, like go down the list, right? And we run all those at high throughput. You get that data back. So you could just use that to pick your winner. If those are just your favorite 1,000 antibodies and you want to pick out of 1,000 instead of the usual thing of picking out of 5, because that's like the more traditional scale. But importantly, you could train a model on that. And then now you could say, well, how would I update my antibody design to be more developable? And that's not a thing you can screen for with a yeast display. Okay. And so that's where AI models can come in and say, okay, here's now a more developable antibody. And so we have a lot of customers asking us to run all those developability assays. And so we're doing those. We're doing thousands a week of the developability assays for people. So if that's the thing, again, if you're in that space, you want to have that kind of data to do model training, I think that'd be great. I also think someone could step in, generate big data sets here, and then be the standard, like make a model out of it and sell the model, right? Like there's just business models here that haven't been tried yet. I don't know what's going to work, right? We know what our knitting is at Ginkgo, which is like complex multi-step protocols run on robotics very cheaply. Like that's what we're good at. And so that's the part of it we're doing. But, people can make all kinds of businesses on top of that. Like people wanting to make a drug for this. Theoretically, other stuff could happen. Yeah.
The first part where you said, hey, someone will send you 10,000 sequences. I'm also interested in generating those 10,000 sequences using language models. I think that's sort of the first part of the pipeline is, how many, one mutation, like let's say ProGen might suggest, 4 mutations, which improve solubility, but then affect binding affinity. So really the goal is to make, 3, 4, 5, number of mutations and then have all of these antibody developability sort of, let's call them points or qualities, biophysical characteristics sort of all improve. Right. And that's where I see, using an ensemble of language models or maybe like an antibody foundational model. I think ProGen is the one that comes closest. So how I'm imagining, and if I want to start my drug discovery company tomorrow, is that I just send like a parent sequence and Ginkgo uses their own in-house model, or, they have I know you guys have some APIs to your own protein language models. and you generate those 10,000 sequences for me and then test it out in the lab and be like, oh, then you can annotate those sequences itself and then feed that back into the model.
Yeah.
And it's sort of that just one closed loop. And I'm just providing like a parent sequence. Like that's it. Like that's what I envision sort of, that's what I thought Ginkgo Data Points is sort of leaning towards.
Yeah, I mean, we can do that. The short answer though is like, people want to, they're in charge of their design. Like for the most part, the reality is the customer of that wants to ultimately be responsible for the model and the design of their antibody and all that stuff, right? Like we certainly have these things. We have an AI team. We can do this, and there are customers we help that with, but like we're not trying to like do your whole
Keep it siloed.
no, like that. What we can do is generate these huge data sets, and like if you did want that, we're happy to do it. But we do see like more interest in just people that want us to make a lot of data, and we're very happy to do that. Yeah,
and I've had a lot of
I would just encourage people like, you can now basically, if you had an area of biology that you thought you could make a living with a model that was essentially a design tool in that area, and you knew that if you had data set X, where X equals some number of, well curated data points in a certain cell type of a certain aspect, and that would make that model work, and you could sell it, then we could just make that data set for you. Like you don't need a lab, you don't need any of that stuff, right? Like that would totally work fine, right? Now the question of can you sell that model to anybody and all these things, but like that's my point. Like, like we really want to just enable people to have the big data sets. And so if there's people also on this that want to talk about like what types of data sets would be of most value to the community, we're also happy to like do a demo in that direction, release it for free, so that if we think more people would then order their specific version of that. So, happy to hear it. Like, I know this is one of the communities of scientists that's like digging in on these problems. What do you want? We also released, there's like a famous paper, Jane et al., that had a bunch of therapeutic antibodies, and we took that whole data set and did these developability assays. That's also for free. So like, what else? Like what would be useful to the community?
Yeah, that's awesome.
So I'd be up for to talk about the other side of the AI house that is sort of like, I think plays into a little more of the like rapid prototyping side of things. And automation side of things, because that's the other area we've been investing in a lot at Ginkgo that I think is like also AI related and worth getting on people's radar. But I don't want to jump off the bio models if you want to talk about there.
Oh no, I think we should, because I know that I'm always wanting to learn about lab automation. I know there's a lot of, I remember at SynBio Beta last year, I looked at Emerald Cloud Labs, also Ginkgo. I did the tour as well, saw the machines. So, a huge fan. I had a lot of friends at Zymergen back in the day as well. So, and, I don't know, a lot of, I think, biologists, I've worked both in software and the wet lab, but I've never used, even an OpenTrons. So I'm interested in learning more about just the role of lab automation in the TechBio industry, which you sort of talked about, just high throughput. You can get a bunch of assays done in like 10,000 assays in like 2 minutes. That is, the value in that is quite obvious. But yeah, I think we'll start off with the role of, Ginkgo's lab automation in the TechBio industry, as well as, how can this sort of help us get to that quicker tools and then move us on to the next phase of developing apps.
Yeah. Okay. So here's where I think like the other half of AI. So there's sort of like there was this like push around, look, the infrastructure to train neural nets has gotten awesome. Let's give more data to neural nets that isn't human language and let them learn to do stuff. And that's your biomodels, and that supports simulation of cells, and that's our CAD tools. And okay, great. So we just talked about that. Let's leave that aside. The other thing that's happened on the AI tech side of the house, like the open AIs of the world and everything, is yeah, wow, we made these language models. That was great, right? GPT-3, GPT-4, all that stuff, 3.5, ChatGPT. But what's really been interesting recently is like if you click the little deep research button on OpenAI and you hit the reasoning model, that goes off and does this like big chain of thought process where it doesn't just like hit the LLM once and run through it and give you an answer. It like it takes 5 minutes to give you an answer, right? Not like 3 seconds. Because what it's doing is it's like hitting the LM a bunch of times. It's going and using a tool like a browser or a Python compiler. Like it is like writing code, searching the internet, and along the way, it's like running through a chain of thought, being like, well, based on what you asked me to do, I think the next thing I should do is this. Okay, I went and did that. Interesting. Based on what I saw from that, I think I should do this next. And it is awesome. Like go if you haven't done it. I mean, everyone on this call should have done it, but like it's the best. It's incredible. Like it's the coolest technical thing I've seen in probably, I don't know, in a while since done. Those things are I think going to be extremely valuable to like go mine the literature and sort of like start to kind of like in company. I think you're gonna are you having Sam from Future House on here at some point? Is that what you're saying?
Yeah.
Yeah, so Future House is like leading the way on this stuff, right? So they're like, okay, we're gonna like mine all the literature and be able to generate hypotheses, okay, right? And like have these models support like kind of like a co-pilot, a co-scientist, like support you in like exploring the literature and coming up with hypotheses and deciding what to do. Awesome. All right. So let's say this happens and now you have a new hypothesis. Now the next step, like traditionally, is that you would go like do some work in the lab and generate some data and like fine. The thing we're exploring at Ginkgo is like, well, could we like have that model be able to engage directly with automation in the lab? Okay, right. And so we have, like you mentioned, Zymergen is a company we in the Bay Area we acquired 3.5 years ago now. The whole reason we bought Zymergen was because they have like the best integrated lab automation in the world.
It's modular as well. It's a bit of like
Yeah.
Lego automation going on.
100%. Yeah, the basic problem, like the reason you never used automation, even though you're a biologist, is number one, the only automation you're really exposed to is like liquid handlers. So like an Opentrons or Hamilton, which is basically something that's doing the job of the pipetting you do. That's only one thing you do. You probably remember going and, oh yeah, I pipetted in this tube. Let's put the Eppendorf in the centrifuge next, right? And then let's flood it down. Yeah, now I got to pipette off the top, and that's headed over to the PCR machine, right? Like, okay, I'm done with that. Like now I got to go to the incubator. Like, it's interacting with 3 to 10 pieces of equipment. It's a series of multiple steps, and like if you just automate the liquid handling, except in a small fraction of experiments, it doesn't really save you much work. Okay, right? Like you still have to do all this other stuff. And so what we've been working on it, and really this was coming out of the technology at Zymergen, but Ginkgo had done a ton of this with other vendors, was like integrated automation, which means the samples move from the Eppendorf, now it's going in the centrifuge, now it's going on the PCR machine, now it's doing this, and you are not in the middle of that as a human. Okay, the robot is also moving the samples between the equipment. That's integrated automation. Okay, an Opentrons or a Hamilton is walk-up automation. You walk up, put some samples on it, does something, and then you pick up the samples and walk them over somewhere else, right? Like you are in the middle of all that stuff, okay, right? And not to mention when you get to that next centrifuge, you better know how to use it because you're the one who's going to go there and interact with the machine and put on the settings and do all the things and don't get any of that wrong. But like you are going to interact with it in a walk-up fashion to tell it what to do. Same with the PCR machine. You're going to put in your PCR settings by hand or something with their stupid interface, right? Like, so all that stuff is being done by a human scientist who has to not mess up any of those steps. All right. Integrated automation means that the automation system is capable of both moving samples between that equipment and then also parameterizing the equipment, like putting in the settings. It can be done with software. So you are not in the middle of that as a human either. Okay. And so that is integrated automation.
It's one big organism in a way.
Yeah. It's one big system. Not a human in that loop. Okay. Right. There's not a human in that loop. And this is critical for if you really want to get scale up and cost down on the generating of the data, like the prototyping and testing of prototypes. So I want to make a thing and then if it's a cell, I got to grow it and then I want to test it and see what happened. And I want that data back to get smarter. That whole thing, if we don't nail integrated automation as a field, we are dead in the water. Okay, it just sucks. Okay, like it sucks. It's too low throughput. It's too easy to take. So you just don't get the data, right? And so there's like cool things happening now with the reasoning models where I think that it could make it easier to connect into this type of automation if you've got the kind of automation systems that can handle like talking to models. And so like we're going to be releasing like a MCP server with like, and this is like this podcast knows, but like, Nerdtown is the tool interface into the reasoning models, right? So now a model can have hands in the lab. All right. That's really exciting. I think that's like the kind of thing we should be doing more of. And I think it opens the door to models basically being able to do reinforcement learning with a lab in the loop. And we're really big believers in that. I think that's cool. We see interest from a lot of people, and that's one of the big things people are buying our systems for. So like, again, if that's an area of interest, that you could be at a research lab, industrial place, startups. I love these racks. They're not too expensive, like, and we can put them in quick because they're modular Legos. So like being able to have an integrated setup that you can run like that, we're trying to make that like easy. Whereas before, kind of like call Thermo, call High-Res, call these like big integrated, they do a custom engineering project that takes 12 to 18 months, not 3. And so that's not startup speed. And so as a result, nobody uses integrated automation. Or if they do, it's like way later down the road. And so it sucks. So that's what we've been working on. And I think that road of like getting the reasoning models to be able to like basically turn a hypothesis into a protocol and execute it and get the data back is really exciting.
Yeah. Bring that to startups. Let's see, I'm starting up with like 4 or 5 people, making a new sort of CDR3, region for her to just random example. How, you said you like you don't want the, what do you call it? The Ginkgo pipeline to be too closed loop because you want startups to have sort of ownership of the project and of the experiment. How does that work in terms of sort of who owns the IP? Is it just, hey, this is our fee and we own all the IP? Is it co-ownership, the model, etc., the data itself?
Yeah. So our model is if you're paying for the data, you own the data, right? Yeah, it's as simple as that.
Got it. Okay. Oh, see, yeah, that works. And you said you're working with quite a few pharma companies right now. I'm curious as to what stage of sort of that drug development cycle, because I know that sometimes antibody developability happens in the later stages, which is an issue, right? Because then a lot of issues, antibody developability is really hard, and the earlier you can sort of get some, positive hits, the better.
Yeah, I mean, so on the developability, like for us, if you're generating these like really big data sets, it's people that want to incorporate developability into their drug design instead of just getting hit by a bus with it later on in the process. Right. So again, like traditionally today, it's kind of like, okay, I went and did this big screen and then I found these 50 and then I did this. I saw which ones expressed well. And then now those are going into these additional developability assays. And like, you're kind of crossing your fingers way late in the process. The question is, if you had like models trained up on a lot of this data, or even just at the beginning, instead of only running your top 5 binders through, you ran your top 500, then you're going to get a more developable asset. And so people are coming to us earlier to do developability than they traditionally would, because with the scale we're at, we can make it part of the drug discovery, and the cost we're at, we can make it part of the discovery process rather than like a thing you run through at the end of your funnel that trips you up a bunch of times. And that's been good. Like people like it.
So it seems like there's sort of 3 different sort of customer bases right now. You've got big pharma, you've got some, startups, and you've also got maybe sort of individual labs and PIs coming to you as well.
Not really. Not like in the academic community. It's tricky. So I think like
there's nothing high throughput there really, right?
We're also. I mean, it's I think where the academic community would be where I'd love to interface more with is like what data sets would be great to have to push the boundaries of like what we can do with CAD, like with computer aided design, with simulation, with like the next AlphaFold. Right? Like those demands should just be made clear. Like we should have that. I wish we had this. I wish we had this. I wish we had this. Like
Be a series of articles we could write. Like, I know we're trying to do a Substack for TechBio Transformersrace.
Great. Like, like just talk to all the best and like, hey, if you could wave a wand and make a data set appear, right? Like, what would you love to build mobs around? Because all the academic community there's plenty of access to supercomputer, like the training models, like all that is there. What isn't really there is the big high throughput labs to generate the data, nor the appetite to spend a bunch of money on outsourced data generation. Okay. But that doesn't mean there aren't people that could spend that money. And if it was clearer to the community at large, what would be most valuable for model training, this is back to the bio models, like what types of data sets, like, let's just be foot forward about it and like get the word out, right? Like and you see some of this, like, Regeneron just did this thing with the UK Biobank to do the so that these things but that, there you go, right? Like that, but that's, one very obvious thing to do. Right? Like, what are all the little niche things to do? What are all, like academics are great at like exploring the full space of biology, and like AI models are going to like, there's other AlphaFolds waiting, right? Like, so like, let's go. Like, what are the data sets they want? I'd love to see what we're talking about. Put it in the comments.
Put it in the comments. Tweet it out. Hashtag, what's the data, guys? And some of the pharma clients you have, my question is, you have your Pfizer's and Moderna, and I visited a Moderna lab before, and, they have a lot of automation. They have the big machines. They've got a lot of money as well, of course. I'm curious, what is sort of Ginkgo's sort of unique selling point to these larger pharma companies? Is it just this, sometimes a smaller team and a well-automated system can get things done a lot more?
Right. Yeah, so I'll dig in one level deeper so you can understand. So what we have with our automation technology, so here's let me just explain state of the art. So state of the art is, you're like, I want to automate this high throughput screening thing. Okay, right? I want to hit this cell line with tens of thousands of chemical compounds. I'm trying to find a needle in a haystack for my drug. All right. And you're like, my preferred assay is this one, and it needs this piece of equipment to do the assay. It needs these 4 pieces of equipment to do the sample prep for the assay. It needs these 3 pieces of equipment to store the samples. So I need these 9 pieces of equipment in a setup. And you call HiRes, call Thermo, like you call one of these like integrated vendors, and they're like, great, let me do that custom project for you. And they're like, okay, well, take the list, and we're gonna make like a 3D model. And we're going to like lay it out, and it's going to take, 12 months, 18 months to like do all this. We're going to semi-customize our software to run your workflow. And then we're going to give it all back to you. And now it's your problem, but it's this like big thing that is like built just for you. Okay. And then the next company calls them and asks for a different big thing just for them. And every time they build one of these, it's like a one-off.
And a couple $1 million, I'm sure.
Oh yeah, that's a couple $1 million always, but also importantly, like, it's fragile to being changed because it was built fit to purpose for the one thing it was being asked to do, or maybe the 2 things it was being asked to do. Okay, this is a very important point, because when then you are a person buying that system, and you're going to spend a couple $1 million and the year of delay and all the stuff, you got to make the ROI, the return on investment for that $2 million be justified by the one protocol that's going to run on that system. Okay, that is a high bar. That is a high bar for a protocol. And so as a result, even in the biggest companies, like the only place you really see in it, not walk-up, not the Opentrons, not the Hamiltons, but integrated automation, no human in the loop, high throughput screening, compound management, right? Like, it's just in these areas that like are very specialized, repeatable diagnostics facilities, okay, where you're just running one money-making thing over and over again. But you never see it in the sea of lab benches where people are doing a lot of variable work. Okay, even if they're doing some semi-high throughput variable work, like they got the multi-channel pipette out or something, if they're only going to do that work for 3 months, how could you justify a automated system?
Yeah.
Okay, right? Like, no way. So what we've done is we basically have like solved that problem by modularizing an integrated automation system so that you can change it easily if you need to, and you can expand it easily if you need to. Okay, like you can make a change to our system in 24 hours. All right, and the reason that's possible is we have, for each piece of equipment, we put it in what we call a reconfigurable automation cart, a rack. Okay, and it's got a piece of hardware, like a third-party hardware, centrifuge, PCR machine, whatever. Yep. 6-axis robotic arm, and a piece of Magnemotion track, which is it's like a maglev track for moving little pucks that have 96 well, 384 well, standard plate format samples can be moved on this track. Okay. And you take that cart and you put another cart next to it. Okay. And you then put another cart next to it. If you have 5 pieces of equipment, I bring you 5 carts with the 5 pieces of equipment in them, and I stick them all in a row. Okay. Right. And I connect the row with a little loop and a place for the cars to park if you want one out of the way. And then that thing can be up and running in no time. Okay. Because I didn't have to design a special 3D model. You just had to tell me what 5 pieces of equipment you want, and I send over the carts. Okay. And because all the carts are the same, they just stick together, then we can have it up and running. And very importantly, if in 3 months you're like, yeah, I'm done with those experiments, but I've come up with a new set of experiments, and shocker, they use 3 of the 5 pieces of equipment I already had. They need the centrifuge again, and the PCR machine again, and the liquid handler. But I'm going to do, this. I need a plate reader now for my output. And I didn't have one in the original thing. We just send you the plate reader cart, and you add it to your system. And now your system, instead of 5, is 6. And you just update, you go in the software and run the new workflow. And what's really cool is if you were still running the old workflow, you could submit both of them to it, and the scheduler will just interleave your protocols, and you can run both on the same system. Okay. And so this is the magic. Like, instead of a automation work cell, which is the term of art in the industry, like this one thing that does, custom and does one thing, we give you an automation core. All right. That you can ultimately use for whatever you need to automate in the future. It doesn't like die on you in 3 years and you have to decommission it, which is what happens right now at these things. It's like, well, time to throw it away. It's like, there goes the money, right? And this is state of the art. And so with our system, you are able to adjust it, expand it. It's really neat. And then the software is like super modern. Like, it's all cloud. You can get the cloud, you can run it on your own internal server if you want, but it's all web-based, and, talks to any of these systems, like I said, including AI models. So that's basically it. But we didn't have, I bought Zymergen, and I was keeping it just for me. So like we like just started, we started selling these like 9 months ago, and we kicked it off at SLIS, and it's going great, but like it's just new on the market. People don't even know about it, but it's that whole, I had it because Ginkgo, we started with 5 of these carts at our site in Boston. We now have 25 in our system, and we've run like 15 flows all interleaved. It's really neat. So anyway, that's what makes it different though. If it for like just traditional automation, and on that side of the house, on the tool side, when it comes to lowering the cost for prototyping and evaluating your prototypes, integrated automation is one of the key things. There's other stuff too. That would handle that, but like integrated automation is one of the big bogeys, and I think this system basically solves it. And so I think everyone should have these. Like, I think it should be like basically standard.
I want one.
Spider-Man.
Thank you so much for explaining this, because it's hard to sometimes find articles written about this. There's a lot of different articles on automation, but in terms of, hey, this is what we need, and this is what the problem is with sort of how we'll be doing automation.
It's because today, like, the automation groups are relegated to just being, like I said, like the hydrogen screening and stuff. So automation is as broad of a topic across all of bioengineering and biotechnology as I think it ultimately will be. Right. Like, and that's because of what I said at the beginning, because of the ROI on the integrated automation has basically kept it in the lab in the corner. And where it really should be is available to every scientist at the company to do whatever experiment they want to do at, say, 500 or 1,000 scale. It doesn't need to be just like the system that runs 1 million compounds a month, and that's, like the only way you can justify it. Right. It should be that you're a random scientist and you want to run 5,000 assays to do a fine tuning, to create the data set to fine tune your AI model, say, and it's custom to your cell line, all this stuff. How do you do that today? Like, you know, like what are you gonna like pull out the pipettes? It's a nightmare. And so right, like these systems would offer that level of flexibility, so more of your scientists would be able to operate at high throughput, which I think is going to be critical as these AI models become more prevalent in our work. So that's sort of the dream, is like every scientist having access to automation at a company, not just the automation engineers in the high throughput screening group. And I think the RACs allow that.
Yeah. And no, thank you so much for explaining that, because I've been reading so many articles and there's a lot of stuff about automation online, but it's just hard to sort of visualize it in your brain as like, oh, it's clicked. I get it. So Jason, do you have like 5-10 more minutes?
Yeah, I can do 5 minutes. Yeah, sure. Let's do it.
Sweet. Let's do it. So we're moving away from bio and AI, but more so on sort of biotech training.
And then within bio, it's a good question. Like now, I mean, look, we started Ginkgo in 2008, right? It was like kind of a time like now. Carnage.
Yeah.
Right?
Through like an iGEM team, right?
What's that?
That was through like iGEM as well. It was an iGEM team and then developed it to come to me.
Yeah. Like we had knew each other. Yeah, we were in the a bunch of the Ginkgo founders were in the first iGEM competition, and like we, yeah. I mean, we were coming out of that SynBio community at MIT. But like the first 6 months of Ginkgo, I mostly like drove U-Haul trucks around, like picking up equipment from failed biotech companies around Cambridge, and like, you know, like getting it for next to nothing. So it's like an interesting time, I think, for people to like take risks right now, like, because you can, you know, like you're not going to get crushed by somebody else in the intervening time while you're like exploring the space slowly, because everyone is like just in foxholes right now. You know, they're not just going to steamroll you. So like, you know, if you have a crazier idea, I think now's a really good time to like take a swing at it. It's a hard time to raise money. That's, you know, that's kind of the other side of that coin, right? Like if it was easy to raise money, then you're probably going to get steamrolled, right? Like if it's harder and you have all the time in the world to go down your little path, right? But like and so like, we spent the first 5 years of Ginkgo like writing grants and kind of bootstrapping the company, but it was during an era where it was like the industry was getting shellacked. So that, you know, I think it, like there's some magic in that, especially for young people, to just like take that risk and go somewhere out on a weird branch of the tech tree and see if there's anything there. Like, you know, you got the time to explore right now, like when the industry is like a little more like beat up. And then, but yeah. So anyway, so I think like, you know, you can go chase down your rabbit hole.
Yeah.
That'd be my
perfect. Amazing. Jason, thank you so much for your time. This was such a fun conversation.